The sheer volume of information that I'm trying to present is way more than a 1 hour webinar on evidence-based medicine. But having been through this process for many, many years as a founder, as an inventor, as the CEO of a company, I realized how much information needs to be integrated in the design process in the very beginning. So that's kind of what we're going to be discussing today. So the webinar outline is in 4 parts. The last week was concept development and ideation, coming up with innovative concepts that meet business models to where your time and efforts can be rewarded downstream. This session 2 is going to be evidence-based medicine, research evidence, and the FDA filing, how that integrates, how your evidence and the FDA filing will integrate together. And I was asked if you guys have questions at the end or during the session. There is the question and answer panel on the bottom of the screen. Also, you can email me at jeff.skiba@gmail.com. I do some consulting with the University of Arkansas and with Arizona State University, but for stuff like this and just to get you started there, I can get you pointed in the right direction at least, and help you with the understanding of where you might be and what might be the resources required to take your product to the next level. What I also will be going over in session 3 and 4 will be how to protect your device or your ideas, so that at the end of the day, when there is an event, and by an event that's either an IPO, that wasn't on my short resume, my little bio sketch, but I did attempt in 1996 to take a company public, which is at the time the .com stocks had taken the market. There's an awful lot to learn about the role of engineering and product selection within that concept, and how to protect what you have in your head and what you're going to be spending all your time working on to come up with something that's marketable, that you can add value into, and you can sell at the end of the day and walk out with a check rather than a pat on the back and a thank you. The grocery stores don't normally take those. So what I will do is go through those in session 3 and 4. This one is going to be on the research evidence and what is really good evidence for your FDA filing. The objective in this webinar is to present the research required for its place next to clinical experience and patient values in order to provide a quality total patient outcome. In the concept of evidence-based medicine is a conscientious, explicit, judicious, and reasonable use of modern best evidence in making decisions about the care of individual patients. Now, this is written for the doctor or the physician who's going to be using what it is that we're designing as engineers. Evidence-based medicine integrates clinical experience and patient values with the best available research information. Now, that's taken from the National Institute of Health article, so there's a link there to that article that was cited. But the idea is that physicians today are being encouraged to go out and find research, and that's where we're at. We're going to provide them with that research. If it's good research, your chances of getting it accepted into the mainstream are much better. And so as you move forward, the evidence-based medicine falls under three very distinct categories: scientific, biologic, and clinical evidence. And we're responsible at this in the design phase of gathering as much evidence as we can under those 3 headings. And very soon into the project, you're going to have to start documenting all the research that you do, and any design of experiments that you may have structured, you will eventually get into randomized clinical trials, and you're going to have to be looking at statistics and biostatistics as far as what is the value of what you've just done. If it's not sufficient, you're going to have to do it over. And if you're under a grant or some funding mechanism, that costs money. So what is good evidence? What is submittable to the FDA? How do you research the literature, and where do you put it? And at the very end, as I put this at the very end because this is part of the evidence that should be presented, are the risk assessments. The FDA started off back in 1974 with a mandate to have truth in labeling. Labels could have anything. Prior to that, labels had anything on there, and it was the kind of the idea of the snake oil. You know, you take a little bit of this oil and it cures everything. Nowadays the FDA is involved not just with the label, but also with the sterilization process, the package that you have wrapped around it, the marketing material, and the website that you put that tells what this device will do. The FDA is involved in all of that, and they are now looking at, this is within the last year, they are now looking at the risk assessments that were performed on the design and what your mitigation process was to each one of those risks. And so the quality system that we'll build around the device will include all of the above. So moving on, evidence-based medicine, you have the basic research, you have the clinical research, and you have the result is improved health. So physicians are trending towards this evidence-based medicine as opposed to what they might have just learned in med school as being a standard of care. So there's a translation of basic science into human studies and a translation of new knowledge into clinical practice. I try to keep that at the forefront of your design process to understand that eventually whatever it is that you're designing will need to be translated into clinical practice. And if you know that going forward, then the way that you document your systems and your evidence will take on a new meaning. You can have a very well-documented and filed system that your marketing people, the people who are going to take over this project at some point and translate it into clinical practice, they're going to need that. And you don't want to go back and have to redo everything at the end of the day. So good documentation practices and filing systems are essential at the very earliest stages. There's been a lot of questions as far as experiments and systematic methods of how to collect data. And what is a good design of experiments? So I went in and I found a very good study under the ASQ. There are a lot of these types of presentations where you can learn more about a systematic design of experiments. In the past, and my first job out of college, that's what got me the job, is that I understood more about design of experiments than the interviewer that was interviewing me. But you need to understand that design of experiments is critical. Especially when you get into the peer review articles and publications that you will be submitting into these different journals that want to see what your design was all about, and that's where you get picked apart. The types of study designs that you may see are pilot studies. Again, everything is documented with a little link that you can go to get more information on this. A pilot study is not a randomized clinical trial, but a pilot study is more to gather understanding of what your device might do before you test it, so that you can have a little knowledge about what the result's going to be. I used to tell all of my engineers that You want to know what the results of your study is going to be before you spend all the money to do the research that you're going to publish, because if you have surprises, then you spend a lot of money for nothing. I mean, not for nothing. You learn what doesn't work, and those usually don't make the most entertaining reading. Meta-analyses. Many people have not heard the term meta-analyses, but I'll go into that a little bit. But this is a way of combining data from many different research studies through a statistical review of those research studies. A systematic review is a little bit less than a meta-analysis, but it is a review of what's out there. Randomized controlled trials. These are studies that are common among the doctors and the physicians that you might be sending this to. They understand RCTs, randomized clinical trials, cohort studies, prospective observational study, case control studies, cross-sectional studies, case reports, ideas, editorials, and opinions. So these experiments are all setting the foundation to be persuasive to the group that you are going to be handing off this research and saying that this product makes sense and that there's evidence behind it. And how you measure this goes from strongest to weakest. And the strongest evidence that you can provide are these meta-analyses, where you take and you look at, over a time period, a statistical review of a set of studies, and looking at, I'll give you for example, in a product that I designed for a company that I formed in 2004 called Fomeris, we had a device that had 200 little micro batteries per square inch. So when it was applied to a wound environment, just putting it to your skin, that the little micro batteries would activate, being moist on the wound, and these little micro batteries would generate a microcurrent on the surface of the skin. It turns out that it wasn't just the microcurrent, but it was also the charge in the electric potential, electrical field that had more of an effect than we could study with the electric current. However, the literature that we were looking at to support this kind of documentation went back about 30 years. And when we started looking at it, it turns out that most of the research was not being reported as being the electric field technology. It was having to do with the electric current. So we were if we would have just studied or looked at electric field technology, we probably would not have had as many hits on our research as we did when we started looking at electric current. It turns out that a lot of the research groups that were looking at it didn't know the difference between electric current and electric field. So that's just a side note, that you have to make sure that your searches are broad enough. But this meta-analysis allows you to go out, and we looked at 30 years worth of research that had been done in this area, and saw that the trend was positive towards that most of the research that was out there showed that the evidence was positive that there was an effect of electric field and electric currents on tissue regeneration. As opposed to the very bottom, which is the weakest, the case reports, opinion letters, we had some of those too. We had animal trials, we had case control studies and cohort studies, but until we got into the randomized clinical trials, did we have any real evidence of what our product was actually doing. This is where your product touches the patient, and you show that it's effective and safe. And these types of studies are the ones that the FDA is used to seeing as being strong evidence in technology. But contrary to that, we should also talk about what's not scientific evidence: YouTube videos, personal anecdotes, gut feelings, parental instincts, some guy you know, many websites, selfies. Take a look, if you have a chance, at www.quackwatch.org. You don't want to be on that page. That's an interesting website. You should also be aware that there are some international groups that have taken over. One is called ASCA, A-S-Q-A, and NICE, N-I-C-E. And I think if I don't have it in this slide set, it'll be on the next one. These are groups who are have been empowered by their governments to look at evidence and present that evidence back to the payors, which are the insurance companies, and saying that this represents the best standard of care for a particular disease or medical issue that our products are addressing. That kind of goes contrary to business and competition because it relies on their research going through this concept of strong to weak evidence. It kind of goes against that, but it is an indication that there are people who are interested in finding out what the best implant. Take, for instance, a knee implant. If you were a surgeon, you would get an opinion from one of these groups that says that the Stryker knee is the best knee to put in for this indication. Not sure how far that's going to go. It hasn't hit mainstream yet, but they are looking at this through this evidence concept. The basics of a randomized clinical trial, if you're not aware of these yet, the randomized clinical trial uses control patients who receive no treatment, a conventional treatment, or a placebo. In a lot of cases, you don't test against a conventional treatment. However, when if all you're looking for is data on how well your product works, that will be sufficient. However, if you are looking at the bigger picture, which is my business, and I'm setting up a business, I would like to have the conventional treatment or a representative treatment also within that clinical trial to make a statement, at least when this clinical trial is published, that our product works as well or better than the conventional treatment and much better than no treatment. There's a use of randomized assignment of the treatment to be tested, and in a double-blinded randomized clinical trial, nobody really knows what that assignment is, so you blind your researcher and you blind your analyzer at the end of the study so that they don't know whether this was a conventional treatment, no treatment, or a placebo, or the device that you're testing. So that is your strongest randomized clinical trial. It's designed to avoid bias, and you'll see that in this presentation a lot, is to avoid bias and masking the reviewers so that they don't know what they're looking at, and you get a much stronger evidence case, and the chances of being able to reproduce this are much stronger. And then development of sound ethical basis for the conduct of the research. Again, you don't want to be putting out research that if someone tries to duplicate this, they don't get the same result. That would be embarrassing to the company as well as to you or your group. These are the fast facts on the randomized controlled trials. And again, you can find a lot of this down at the bottom here. This is a Medical News Today article. And randomized clinical trials are considered the gold standard of clinical testing today, especially for new medical interventions, and this applies to drugs or to devices. And just to back up just a second, this slide deck is more designed towards medical devices than for drugs. I don't spend much of my time in drug development. I have a little bit of understanding of what goes through to do a new drug, but not enough to give any kind of advice on that one. But on the medical devices, class 1, 2, and 3, which I'll get into in a second, is that I have been exposed to all of those. So Randomization means pure chance is used to decide who gets the treatment and who goes in the comparison group of the trial. And a randomization is done so that selection bias is removed. I did a study at the Walter Reed Hospital in Washington, D.C., and we were on a randomized clinical trial, and out of 60 patients that were scheduled to be enrolled, the first 5, 3 were controls and 2 were treated, and then we found out that the oversight committee had consumed all the research dollars, but it shows you that sometimes when you're doing this selection, you're removing the selection bias, that you could end up testing all of your, or most of your control group at the beginning, and then all of your treated group will be at the end, or most of it will be at the end. The placebos may be used for the control group, and let me talk a little bit about placebos because this is, it sounds easy, but these are dummy treatments that look the same as experimental treatment but don't contain anything. You need to be aware of these, that these placebos also cannot contribute to anything. So your design of your placebo is important. If you're putting a placebo in, just the fact that the placebo has been introduced into the system may have interaction. So a good placebo design needs to be documented and performed, or you could have a false negative, or a false, and in this case, the placebo is supposed to do nothing. You may have a false result. So it's also not always ethical or practical to give a placebo, such as this would mean denying treatment to people who have a life-threatening or serious illness. I went through this with the evidence-based research group in downtown Phoenix, Arizona, where we were talking about a group of patients that we wanted to test our product on. We were looking at the potential for eliminating amputation. And the question was, if we thought that our product could eliminate amputation in a patient population, how many of those patients that we put into the test should we allow to go to amputation? And at what point do you say that this patient is going to be amputated? These were all ethical questions that were brought up, and the fact was, and it was our decision, that we would not deny treatment to any of the patients. As a matter of fact, if you took 30 patients, which 30 is usually a pretty good number for these types of tests, just FYI, that if I took these 30 patients that were scheduled for amputation, and I made my criteria that they must have been scheduled for amputation before we apply our product, then what are the chances that a patient scheduled for amputation at some point would just miraculously cure itself and not end up being amputated, and you wind up with a statistical confidence on that? And the answer is, anybody who's scheduled for amputation usually gets it amputated. So there's a humanitarian aspect to this and an ethical aspect, where if your treatment might cure this, you may have to go in and increase your number to cover the number of spontaneous miracles that cured themselves without any intervention, and the fact that you did not include them in your treatment or your study design. So in getting into the study design, why do the study? Why are you studying it? What's your hypothesis? Your method's very clear. Your sample size with statistical confidence. This is probably the most difficult aspect of this. Not enough patients. You don't have any. You don't have a statistical foundation. I ended up having to bring in a biostatistician to help me with the work that I was doing at Walter Reed and with the Department of Defense on statistical confidence levels. In my randomized trials, But the rationale for going down this is that that your sample size is defensible in this randomized trial to minimize overall trial sample size. You can do this obviously with a pilot study to help set that statistical confidence interval, and then through that you're going to provide credible results. If you're going to be critiqued on your study design, this is where you're going to see it, is in your statistical confidence levels and what your rationale was. Are your results persuasive? And do they support your claims to the FDA? And I'll get into that in just a second on the claims to the FDA because that is that's a loaded statement. In clinical relevance, does it support my marketing and sales collaterals? And that might be a new term for some. A sales and marketing collateral are the brochures and the pamphlets, the handouts that you use to introduce your product to the user. In this case, a physician, a nurse, a hospital, a PA, whoever's going to see your product, who's ever going to introduce it to a patient. Then you're going to have the user, the patient who's going to have the product used on them or in them, if it's an implant. And if you have this research in such a state that your marketing people can't understand it, they can't use it. And it's important that if it's good research, that it be shared with your market. And so that's where I put this clinical relevance, that you want to be able to pick out the pearls and the gems that are inside this research and say, here's what this really means. And coming from the research group, it makes a lot of sense to provide that clinical relevance within the study discussion. In addition, you need to be aware of protection of the patient privacy. They're in your study design, and when you're looking at the patients that you're going to be including in your clinical study, you have to be aware of the HIPAA laws, and so I put that link to HIPAA in there. But the thumbnail of that is you can't share patient information without their consent. You must have that written into your research in order for the research to be approved through the reviewing organizations, usually IRBs, investigational review boards, or institutional review boards, whichever you want to call them. But these are groups that will review your studies and put their stamp of approval on them, so that you can move them through the hospital or through the research group that you're using. Now, I'm talking about the top level clinical research first. We can back up and talk about the study designs for biologic research, as well as the scientific research. These are also required to have some kind of a statistical rationale behind them. If you're using laboratories to do your animal research, or the benchtop research, if you're going to be submitting this data to the FDA, if GLP is available, this is good laboratory practices, that you submit with good laboratory practices. Also, it may add another $100 to $200 per study onto your test budget, but it is important if you can get it. Otherwise, you're going to have to make some rationale in your submission, in your FDA submission. The clinical evidence, the hierarchy of evidence, I put down these two good links with the National Institute of Health and the AHRQ. This was one of the groups that's reviewing this types of data. But this is a good website to see where the market is going as far as who's accepting what type of science. Your evidence files then become critical in your product design and your product design control. The highest risk to a project is the loss of good data through poor documentation. This happens all the time, and it's something that if you can put this on your desk, but this is the from a management perspective, we spent a lot of money to get this data, and now I can't use it. That's kind of what that says. It's because it was poorly documented. I've seen studies where the design of the experiment was so rigorous and so time-consuming that the study the study controller, the person actually doing the study, had to take so much time away from their practice or from what they were doing, that it became impossible for them to fill out the study forms and fill out all the data that we were asking for. And so we had to go back and reset that data sheet, minimize the amount of interface that's required with your researcher, so that you can get the data or get some data. Some good data is better than a lot of bad data. So organize the evidence. In the beginning, you may only have a few case studies. Case studies are a little bit different than these randomized clinical controls. This is where you use your product on a case and it worked, or it didn't work, or it might have worked. But you do want to have a few case studies. From a marketing standpoint, these, if in the beginning you're marketing these products to your funding groups, and these are good evidence that you're on a track to some kind of success. But your objective is to build that evidence file and to have it in a location, whether it's Dropbox or Evernote or some other system that you use, so that it's well organized. And third party evidence is also very good evidence because it confirms what you did. And by third party, I mean somebody other than you performed the evidence. They repeated the test, or they confirmed the test, and they eliminated the test bias. And you know, even though everyone are upstanding, honest people, there are still people out there who think that since you did the research yourself that you introduced bias into it. So this is a way of confirming it. If you have the money, or you can budget that in to your study design budget, then you can put in confirmation and eliminate that bias. Going back into the clinical and economic benefits of the work that you're doing, this should be designed in part of your discussion. In your research, you look at the clinical economic benefits as part of the discussion section, looking at how much training is required for someone to take a look at this product and understand how to use it. How to use it effectively. You need to be aware that some of these devices will add cost to an already capped medical procedure. So if a surgeon is getting, let's say it's a knee procedure, a total knee procedure, and they're removing the implant from the knee, or they're putting in an implant, that could be $3,000 to $3,500 capped, and that means that's got to include the nurses, the operating room, and the doctor. How are you going to add more money into a capped medical procedure? So there are ways to work through that. Proof that the device works better than today's standard of care. That's why I go back to the study design. Put in that one into the study design. Put the standard of care into the design so that you'll have that data ready. Reduction in cost. Something that you might not always have in your study design is that the device will result in a reduction of cost, and cost is again a very, very big umbrella. There's a lot of things that introduced cost. An amputation today costs between $95,000 and $100,000, but that is a rationale for eliminating all of the advanced wound care techniques that doctors and nurses are doing within the hospitals to reduce the cost of the treatment, because some of these wounds that are considered as in the advanced wound care stage, they're considered to be chronic wounds. They have already consumed close to $100,000, so they would rather spend $100,000 to stop the bleeding, so to speak, and reduce the cost because, and this is the next line, because they can show a reduction in readmission rates. So if you have a patient that keeps coming back, there is a cost associated with that readmission. Now, whether that cost is due to infection, or because the device failed, or because the patient was non-compliant, doesn't matter. But your study design you can throw this into the study design and have the answers, and not have to go back and redo. Redos cost money, and if the research contributes to individualized medicine, that is even that's the icing on the cake, because this is where again where medicine is trending right now. Preparing the evidence for filing. For those of you who have not been exposed to the FDA, there are two main classifications in the FDA: devices and drugs. There are some subsets to that which are now being more accepted. However, the FDA has not completed all of their guidance documents, but the subsets are de novo devices and combination devices. But within the two umbrellas, drugs or devices, if you're not sure what that is, there's a submission which we call the 513G that you can submit to the FDA, asking them to look at your product and decide for you whether it's a drug or device. And if it falls within the devices, it's under the CDRH, which is Center for Devices Radiological Health, and your device will be then classified as either a 1, 2, or a 3. If you see the slide, general controls, special controls, and general controls, special controls pre-market notification. Class 3 is the most expensive and the most time-consuming. It's basically saying that this device is a life support or life-assisting device, or failure of the device could result in serious harm to the patient or death. Class 2 devices are general controls and special controls, and Class 1 devices are just general controls. Class 2 devices are your 510K type devices, and about 40% of the devices that are marketed today fall underneath that class. About 55% fall under Class 1. So the very first thing that you want to do is classify your device, because in this process of providing the evidence for the filing, you really cannot market a medical device even if you state it's not FDA approved. I mean, this is just an invitation for a knock on the door, and the FDA has the right to shut your company down, and that results into well, one, if your company is shut down, it's going to be tough to raise money around that. So this is an important slide to understand that these devices do take on a different life once you have classified them. So this slide was taken from the FDA.gov site. Step 1 is to classify your device. Choose the correct pre-market submission. That's either 1, 2, or 3. And in the case of a 1, you don't have a pre-market submission. You only have a 510K or you have a PMA. Prepare the appropriate information for the FDA, and there are guidance documents for those, and send it to the FDA and interact with the FDA staff during the review. And they've actually today they've allowed us to do what's called a pre-submission to make sure that the evidence that you're going to provide is adequate. It takes a little bit more time, so if you don't have the time, you might want to just consider following the guidance document to the letter of what the guidance says, and then send that in, and then complete the establishment registration and device listing. Getting into the classify the device, under the 510K section, I'm going to go down to this line right here. It's the fifth bullet point. It has the same intended use as the predicate. This is where people get hung up. The 510K by itself is a demonstration of substantial equivalence. Substantial equivalence means that the new device is at least as safe and effective as the predicate. It doesn't mean that it's equal to. It just means that you can demonstrate that its safety and efficacy under the same intended use as the predicate. If you change that intended use, understand that the FDA uses, in this system, in the 510K system, they use decision trees. And if it does not have the same intended use, it will drop into a different branch of the decision tree, and you will probably not be given your 510K until you change that intended use. A device is substantially equivalent if, in comparison to the predicate, it has the same intended use, same technological characteristics, or it has the same intended use and has different technological characteristics, and the information submitted to the FDA does not raise new questions of safety and efficacy. So this is a very important slide. It means that the claim of substantial equivalence does not mean that the new predicate devices must be identical. However, substantial equivalence is established with respect to intended use, design, energy used, delivered materials, chemical composition, manufacturing process, performance, safety, effectiveness, labeling, biocompatibility, standards, and other characteristics as applicable. So when you're looking at study design, whether it's clinical biologic studies, this is the kind of evidence that we have to provide, that the materials, the evidence to support the material, the chemical composition, the manufacturing process, all of these have a study, or at least the risk associated with this particular aspect of it, that we have looked at this and we have decided that this has no risk, or it has some risk, or it has a lot of risk, and we're going to control that through a quality system. The PMA, however, is a little bit different. Now, this one's and I've been in both, and the PMA is a tough road to go down because there is no information on these devices to generate performance standards under Class 2 or general controls Class 1. So you're really going to have to provide the FDA reasonable assurance that the device is safe and effective through your study design. And if they've seen it before under the 510K, it's a lot easier. So if you have the chance to amend your indication for use, you can go that route, reduce the amount of evidence that you have to provide. If you decide that you want to go this PMA route, these are much more expensive. I'll go back to the 510K. This route, anywhere from $10 to $24 million is about the average it takes to get a 510K today. Through a PMA runs into the millions and millions of dollars. I mean, this is the last one I did. We had put in over 200 million, and we still weren't approved. So when you're talking about clinical studies, I didn't talk about the amount of money that goes into these. If your clinical study, especially the randomized clinical trial, if you're talking about the cost per patient, they could run anywhere from $500 per patient to $30,000 per patient, depending upon how long you have to watch the patient or the animal, for that matter. How long you're going to have to keep that animal or patient under observation, and how much interaction your researcher is going to have with that animal or patient because of the study design. So if your study design tells the researcher she only has to interact one time, that's a different than one that she has to interact 100 times. So you can see that the money aspect starts to come into play. But the PMA is like an approved new drug application. It's a private license granted to the applicant for marketing a particular medical device. A Class 3 device that fails to meet PMA requirements is considered to be adulterated under Section 501 of the Act and cannot be marketed. Pre-market approval requirements apply differently to pre-amendment devices, post-amendment devices, and transitional Class 3. This is a very difficult road to go down and requires a commitment. But you should know that the devices under this 510K are not approved for market, they're cleared to market. And the assumption is that under the 510K, your quality system is already in place. So a medical device that meets the 510K requirements, you have defined the packaging, the labeling, the sterilization method. You've tested all of those. You've tested your manufacturing process is set, your design is frozen, and your quality system is in place. And if those are not met, you still cannot market your device. If the FDA usually does not come in and audit a 510K product right off the bat, but usually within a few years they will be into your site to see if you have that in place. If it's not in place or you're deficient, you will either get a 483, which is a report of deficiencies, and you'll be given some time to correct those, or you'll be given a warning letter. And either one of those can harm your ability to raise capital. It will definitely, if you're a public company, will definitely affect your stock value. So all of these have consequences. So the 510K is cleared, not approved. The PMA is approved and allows you the license to market the product. The downside to the 510K is that if there are other devices out there and they've already been cleared under the with a reimbursement status, that their reimbursement under the CMS, the Medicare group, the Centralized Medical Service, they have established a code for this, a classification code, and you will use that classification code in your 510K, and when you go to the CMS, they will want to see what classification code you were cleared under the 510K, and they will give you that same reimbursement going forward. So if your cost of goods and the cost that you're going to have to charge for this is substantially higher than your competitors, all of a sudden now you can't get reimbursed. Somebody has to pick up the difference between that and whether you charge the patient itself, like in cosmetic elective procedures, or whether you have to put it onto the capped medical system. Those can make a huge difference in how you decide whether to go a 510K or the PMA. See, the PMA process allows you, because you're setting the classification, you're going to the CMS is going to see this as a new product, and they will work with you to assign the reimbursement amount for that product. As I said before, the FDA uses decision trees, so I left this in. Again, down at the bottom you see this presentation is worth watching on this, or going through this presentation. It's on FDA overview. But if you look at this path for a path to get to the product to a patient, the new device is compared to a marketed device. Does the new device have the same indication statement, yes or no? If it's no, look where it goes. It's not substantially equivalent. So what you want to do is follow the path that says the device is substantially equivalent. Now that by itself adds value to your company. Once you get to this point here, the device is substantially equivalent. That is a value add to your effort. Let me go over to this one. This is another one. This is a pre-amendment device. Is the study exempt? Yes or no? Is it an invasive device? Does it have significant risk? So this is what the FDA uses, and every one of these devices that you might be considering, the first thing you do is you go and you search to see if you can find guidance documents and decision trees to help you decide which tests you're going to need to run. Preparing the FDA submission, I put there's the guidance document link right here for regulatory submissions. Literature searches, you can go through, you can look at freedom of information on the FDA site. If you're doing a 510K, you're entitled to see other 510K submissions. However, they've been redacted, so you'll see a lot of little black marks on them. You won't be able to see everything. Indications for use are going to be determined by the time it's exposed to the tissue and what the raw materials are used. Those are the two big ones. Whether or not your classification is going to be determined upon this: indication for use, time exposed to tissue, and your raw materials use. So stay in the category that they've seen the raw materials before, and that the time exposed to tissue is explained. That's not a 510K killer. Your indication for use can move you around a lot as far as what the evidence is that you're going to have to supply. The studies that you'll be involved in, biocompatibility is a big one. I mean, almost every implantable device, any device that touches the skin that stays on the body for more than 24 hours is required. You have to do some form of biocompatibility, and this is an iterative approach. Many times it's best to bring in a chemist and a microbiologist to look at the biocompatibility that you want to do on the device and develop a rationale. You can save money by eliminating some of the tests that are required early on, but that does take. It's not that you can just say not applicable. You have to give a rationale why you didn't use it. Foreign clinical study results can be introduced into the FDA under certain conditions, and what happened here? Okay, under the 510K, the FDA user works off the decision tree, find a predicate device, and use comparison charts. The PMA, the company and the FDA will meet. You'll have to get with them because they have PMA coordinators, and you'll have to go prepared with a plan. Don't let them tell you what you need to do. Go there and say, this is what I intend to do, and then defend it. There are fees associated with this, and there's the ability to do what are called pre-submission meetings if you're concerned that your 510K or your PMA evidence plan is not sufficient. Here's the typical fees for the FDA. I always fell under the small business fee, but you can see for a PMA it's considerably more. For a standard company, it's almost $235,000 just for the submission fee, compared to the 510K, which is about $5,000. So, but you can use that. And again, look at the if you need that link, the link is included. Registering with the FDA, I use this slide again because it's important that you understand that you must be compliant with the FDA's standards. The basic testing overview, taking a look at the material characterization, toxicology, biocompatibility, sterilization, validation. I introduced these here because these are in addition to the biocompatibility and toxicology studies. If you're going to sterilize your product in a package, your package integrity needs to be tested, as well as your sterilization method. If it's there's only a few of them. There's E-beam, gamma, ETO. Some are doing plasma and steam. These are your choices, but your package makes a difference. If your sterilization process destroys your package, you can see that that's not a good idea. So you have to test it. I had an experience back in the early 90s working on a cardiovascular product. We had put the product into a double tray. The inner tray and the outer tray both had lids. So you put the product in the inner tray, seal the lid on top, and then put it into the outer tray and seal the lid on top of that. That all went well. But on the trays, they decided to print the label rather than putting a stick-on label. And when they printed the label ink interacted with ethylene oxide, and that ethylene oxide ink combination created a toxic substance. So we had to recall 5,000 units because somebody in the purchasing group had changed the ink for our printer to print our labels, and the ink created a toxin that we couldn't allow to remain in the pouch, because if they opened it up, the nurse got it on her hands, and then she handled the device goes into the patient gets sick, and now we have to do a medical device report, which is an adverse event because our ink was bad. So potential biocompatibility studies, keep this list because these are the common ones that you're going to see, and you need to consider each one of these. for your device if your device comes in contact with the patient. Risk assessments today drive the quality system. ISO 14971, and I'd give you links to these, but there are no links to these ISOs. There are some explanation to international standards, but they require you to buy these. And the FDA will give you the guidance documents for free. The ISO, you have to buy them and pay for your copy. Learn the failure modes and effects analysis and PFAMICA, which is the process failure modes and effect. So while you can look at the device and you can say, here's how it will fail and here's my mitigation for the failure, if you're looking at the process on how you made it, you have to go back and say, here's how my process might fail and I could get a defective result coming out of it. Documentation of this risk assessment means probability of occurrence, seriousness of the failure, and how you mitigate the possibility for failure, either through design or through education or contraindications. ISO 14971, I just put down kind of a summary here of what they require under the 14971. It is very similar to the FDA, so it requires us to go through each one of these elements of the risk associated with every aspect of the device. Build a portfolio, know the standards, build a device master record, build what's called a design dossier. This is going to be everything that you've researched, everything that has to do with the safety or efficacy of this product or your submission. Anything that I would put in there, all your marketing stuff as well, anything you found out about the market size in your research. Follow a quality system early on. A lot of those are available. However, the new requirements just came out, so it's changing. Be aware of that the quality system requirements are changing. And keep manufacturing, that you're eventually going to have to put this into a manufacturing process, and the requirements for your risk also drop from your product into your manufacturing, and understand the effects of the cost. I included some more helpful links here, the Food and Drug, International Standards and Design of Experiments. So these are some good links for later study. The FDA provides a nice website here, or CDRH Learn. It's an online tool. It's available 24/7. But here's some of the modules that they have applied, and I would highly recommend, if you're going to be going down this road, that at least get a working knowledge of each one of these. And I thank you for your time. I think my time is, yep, I'm 2 minutes over, but I hope I didn't have to rush too much through this. I will turn it over to questions and from the group.